also help us lead to some of those medicinal applications such as antibiotics. And bats. I think people kind of underestimate the ecological role of bats. I don't like mosquitoes. I don't know about you guys, but if bat populations started to decline, then we can see some serious incline of mosquitoes. If you look at that, one bat can consume up to 1,000 insects per hour. And like I said before, these bats can also affect the microbial communities of the caves in which they're in, because there's lots of evidence of guano in these caves. Another thing is white nose syndrome. I don't know if you guys have heard about this. It's a fungal infection, and it's caused by a fungus in the genus Fusarium. And it's currently in the northeast region of the United States, and it hasn't been reported down this far yet. But it has a 80 to 90% death rate in infected bats. And as you could guess, that could be pretty devastating to a population pretty quickly. There's not a whole lot known really about this right now. I think they're really still working on it. In Alabama, they have ordered the closing of bat caves on all of the national forest land, I'm pretty sure. And that's to try and help stop the spread of this disease. And if it does come down here, if those are closed, then that keeps people from, possibly spreading it from one cave to another. And the bats can be highly migratory. They can migrate hundreds of miles. So just bats alone spreading this disease could cause it to spread pretty quick. And again, like I said, there's very little known about this particular disease. How is it entering the system? How is it being allowed to flourish? Some of the only things I could find out about it is that they were suspecting that the cause of death was that these animals were waking up during their hibernation and then overburning their fat supplies and not making it through their hibernation period. So now for this the methodology here. As Dr. Barrons said, it was kind of interesting watching him inside the cave here. But he came in and helped out. So we took these samples, and they're from the top 2.5 centimeters of the soil. Now what we did is we took samples from a shelf. There was a fairly distinguished shelf, about, say, 3 feet, about waist height throughout the entire cave, and then the floor. So what we did was we took a sample from the shelf and a sample from the floor, and we pulled those two samples together about the same weight of those two samples together to perform an analysis with. So when it came to DNA isolation, there were several difficulties that we had with this. The first thing we tried was we tried a commercial kit, and had very limited success with that. And then we tried some various protocols that were in papers, including the use of PVPP, which will remove humic acid. Humic acid is common in soils, and what it does is it, most of these DNA isolation techniques, humic acid will separate out with the DNA, which will then in turn cause an acidic environment and not allow your downstream reactions such as PCR to work. And also, there's fairly low concentrations of DNA in these soils. You're thinking we're not taking a tissue sample, for example, from an organism and then grinding that up and trying to get the DNA out of it. We're taking a sample that's soil and trying to get a small amount of DNA out of that. So the kit, we actually found a kit that worked, and it was this particular kit from Mobio, which the first kit we tried actually was a Mobio kit as well, but it was one of their, I guess you could say, less expensive kits. And I guess they get what you pay for when it comes to some of these things. But this one worked very well, and it wasn't too bad. But so when it came to amplification, we used the 16S ribosomal DNA, bacterial specific primers. The 16S unit of DNA is a highly conserved region, but so it lets us be able to do comparisons with it, but there is still enough variability within the gene that we can determine differences between organisms. Those are the particular primers that we used. And we ran this out at 25 cycles. We want to keep the cycle number to a minimum when doing PCR, so we kept it at a pretty low number there. Then we set up clonal libraries. Basically, we took this section of amplified DNA and then inserted into a vector, which is a plasmid in this case, and then into the bacteria, where the bacteria then kind of do our amplification for us with the subunit, and then we extract the plasmid later from the bacteria, and that's what we'll use for sequencing. And as you can see, here's a gel, and this is a 1500 base pair segment from 16S. And here's a gel showing several sample sites that we took. Now, we only used one full sample from one site in there, but the other samples are there. If we could ever get back to get some, they're in right in the region that we wanted, and that was good. That's a pretty clean sample there. And sequencing. So we use the background culture sequencer, which is a very interesting little machine down there that you have to talk nice to get it to work. Anybody that's used it before knows you have to sweet talk the sequencer. But we just use forward priming for these sequence analysis. So they're partial sequences. They're not a complete sequence of the 16S gene. We selected for a minimum length of 500 base pairs, and then these sequences are compared to null sequences from the CEPI base. And they're sort of then we went into analysis with this software suite, Mega 4.0. This set of software, it contains materials that let us do a lot of different things. It's sort of a compilation of a lot of different analysis tools into one software suite. So it's a lot easier to use this one particular one than trying to cross between several different programs. And with this, we performed alignment and trimming, which I'll go into a little bit of detail of how that works and why that's important, as well as phylogenetic analysis, such as statistical analysis of bootstrapping. So at the top here, this section right here is kind of what you get as soon as you put your sequence in. You've got this kind of mess of nucleic acids. It doesn't really mean a whole lot. So first thing you really want to do is you want to get these aligned and see if you look at, like right around here, we have a lot of conserved regions throughout these, and that's what this does. That's what the alignment is put these sequences together and sort of helping us make sense of what's actually there instead of just having this mess of information up here. And if you look at the bottom here, this is the purpose behind trimming. This is the forward end of a set of sequences. Now it may get a couple reads extra on one sequence than another, and if you trim it, then at that point you have one section of aligned DNA to do analysis on. So it's a lot easier to get the looks of confusion. So after doing all this and after looking through what we had from the